questions that people have over the material. If there are not questions, then I will just go over highlights, the best of. Does that make sense? And then if there's time left, and you guys ask questions as we go, if there's time left, there probably will be that many. Then we can talk about next class period is your see you again until minimally February. We need to talk about, I'm not going to say at least February. I'll probably only be, well, only on Tuesday for certain. Not. But you're going to have to go to study hall. We'll talk about it. Let's get through the immediate. We'll talk about it.
So, I'm sure osteoclasts.
Yeah.
That means, what do they? Is it? I know it's.
I have those slides. So we'll,
okay.
That's a good one to
yeah, I just.
Are there any other areas of concern? So she wants to hear about osteo.
That's like the big.
So the class
Building. That's right. Okay. So first, I wanted to talk about negative feedback. All right. What negative feedback was? It's. Do you guys want me to go over the abstract parts of a homeostatic system again? Hi, Sam. You're cool. So, huh? So, let's go to this slide. Hid these ones. Okay. Basics. Let's pretend for a moment that, let's not worry about feed-forward controller. I'm not going to ask any questions. Okay? Feed-forward controller, it's like anticipatory modulation of a... That was the example I gave. But there are many other feed-forward controlling, but it's not to us. In general, there's some sort of, the thing to start with is the regulated variable. Hey. What was I saying? We start with a regulated. And that can be anything, any kind of physiological body temperature, blood sugar, the amount of carbon dioxide in your blood, like any sort of thing that's measurable in your body is likely to be some sort of regulated variable. Something that's measurable that can fluctuate is going to be some kind of a regulated variable. And there's going to be mechanisms, sensory organs, sensory neurons, that are going to monitor sensors. Those sensors are going to send their information to some kind of integration center, maybe in your brain, maybe in some sort of endocrine tissue, that's going to take that information and essentially compare the value of the regulated variable to a set. So This is what the reading says my body temperature is, and this is what I want it to be. And so this means I'm below that set point. I'm going to do things that are going to raise my, or my body temperature is too high. I'm going to do things to bring it back down. That integration center makes a decision, right, by this comparison of the set point to the value of that regulated variable, and then it sends stuff out to an effector. Some sort of thing that's going to affect that regulated variable, cause an effect in the body. All right, so do this. All right. So here's an example of homeostasis, Gesundheit, in a type of homeostasis called negative feedback. And I want you guys to understand the definition of negative feedback, what negative feedback actually is. Negative feedback system is one in which the response of an effector, whatever that effector is that's causing a change in the regulated variable, is going to oppose the stimulus. So if this is our set point, and the sensor says our body temperature is low, we're going to use this negative feedback is going to engage effectors that are going to oppose the stimulus. The stimulus says our body temperature is going down. We want an effector that brings it back up. It's going to oppose the stimulus says, oh, we're getting too cold. Let's oppose that. It's negative. It's called negative feedback. All right? And it's called negative feedback because so here's the integration center. Here is the regulated variable. Here's the sensor. Here's the effector, right? Which affects the regulated variable. As the effector happens, as this is sending signals in, this signal that's coming from the regulated variable gets less, right? Because the regulated variable is coming up to the set point. It's approaching the set point. So the effector goes down. The amount of the effector goes down. It's like when you're coming to stop, you press on the brake and the car starts stopping, and then as you get close to being stopped, you're very lightly. So the effector, as the effector works, it needs to work less, right? So it's this sort of negative feedback system where the system will eventually come to rest, stop at hopefully the set point. So, for example, the important words here are the response of the effector opposes the stimulus. That's the important little. We have a thermostat. This has our set point that we put in it. This is the set point. We set the thermostat to a temperature, and I guess we'll start here. This is the room temperature. We have a thermometer in the thermostat that measures this. This is the sensor, sends information to the integration center. This sends it out to the effector, which is the air conditioner or the heater, whatever. And the temperature wavers up and down around that set point. Negative feedback. In the body, it's just the same. Where there's body temperature, we have thermal receptors in the skin that send information to the brain. That's the integration center. And then it sends signals out to various effectors. There's sweat glands, you shiver, et cetera, or you flush, your capillaries dilate. All right, negative feedback. Here was another one that we talked about. A postural change in blood pressure. So down, get up, blood drains from your head, raise the blood pressure. So the baroreceptors, the pressure receptors in the blood vessels that are above the heart in the aortic arch and the carotid bulb, they send signals to the brain that blood processes that information, and then signals from the cardioacceleratory region of the brain goes to the heart, speeds up the heart rate, and then that raises the blood pressure. Don't black out. All right. Another thing that you'll want is the mode of communication. So we know that there has to be communication for these homeostatic systems, and exactly what kind of mode of communication is dependent upon these three factors: how far away the integration center is from an effector or whatever it happens to be that is being regulated, so distance. Speed, how rapidly it needs to happen. Is it just like is it a long-term growth homeostasis, or is it a rapid response to something? Or is it somewhere in between? So the distance, the speed, and then this idea of distribution. Is it like really tightly focused? Is the effector just one specific muscle in your eye or something? Or is it your whole body? Distance, speed, and distribution dictate what kind, whether we're going to use the endocrine system, chemical system, the blood, or sense, distance, speed, distribution. Yeah?
Are we going to need to switch? Use to communicate slowly.
I am highlighting the information right now. So like when I'm talking, like the things I'm saying. So I didn't actually focus on that. So that is something I talked about, but I'm not focusing on it. I would not spend all my time worrying about that. I talked a little bit about how, then we moved on to talk about calcium homeostasis and the ways that calcium is used in the body. I talked about it in terms of exocytosis, the release of vesicles. That this is like how a cell would release some chemicals into your bloodstream. This process of exocytosis uses calcium. Calcium is essentially the key that causes this fusion. Calcium is important in the blood clotting cascade, muscle contraction. Yeah, oh yeah. That's right. Well, in fact, sweating does involve. Yeah. What you sweating is exocytosis. What happens when you sweat is you have these sweat glands that are like this, right? Huh? You don't want that? Well, you have it, and you would do well to be glad that you have it, because I worked at a camp for children with chronic skin disease, and they did not have. Some of those kids did not have sweat glands. That is not. It's really not fun. They have to always be careful about what they're doing. If they overheat, they have to get in tubs full of ice. Yeah, dishydrosis is what that's called. It can be life-threatening. Anyways, this is a sweat gland. These are ducts, D-U-C-T-S, ducts, that the sweat is going to come out of this onto your skin, right? But this little chain of squares that I have along there, those are individual cells. And those individual cells, you can imagine this is a zoom in. This is the surface of one of those cells. and the sweat is coming out by exocytosis. So each of these cells is doing exocytosis when you are sweating. And that sweat goes into a sweat gland and comes out onto and evaporates your body. Exocytosis, its definition is pretty well described by its etymology. So exo, meaning outside of, and cytosis, the cell, and osis being process, cyto, cell, osis, process. It's the process of sticking stuff outside of the cell. It's just it's the process of membrane fusion of a vesicle with the outside membrane. That's all like way, but that's what really important function of primarily endothelial cells, but also neuronal cells, exocytosis. This same process of exocytosis is involved in nerve cells for neurotransmission at the synaptic bouton, at a synapse. So the way nerve cells work is you have like the cell body, which would be like your body, and then there'd be an axon, which would be like your arm, and then they have the telodendria that stick out from it, and the ends of your fingers would be these synaptic knobs. And then at the end of it, you would have a little bit of exocytosis. So like, for example, I would be synapsing on you. You would have some receptors on your fingers, and I would be releasing neurotransmitters via exocytosis. Receptors on your skin would be picking them up, those receptors up, and then causing this cascade of membrane dynamics that would happen throughout the neuron. Sadly, we would probably not talk about it. It's really so. For example, one kind of a neuron that is in your body is these are neurons that use serotonin, and serotonin is a neurotransmitter that is often associated with feelings of well-being and formation of memory and stuff like that. All of the things that your brain does that you like. And so sometimes people have, when the neuron releases that serotonin, this is not going to be on the test, I'm telling it to you because now or never, you'll never hear it otherwise, I suppose. The serotonin gets released and causes a downstream action potential in the next neuron. But then this upstream cell recycles all that serotonin and brings it back up into the bouton to be used again, right? And sometimes some people have a problem in their cells where this neuron is not making isn't making a whole lot of serotonin, or is like really too good at recycling it back up into the synaptic knob. And so that it's not on the downstream cell long enough for there to be the response from this cell that there should be. So they have drugs that maybe you've heard of called serotonin reuptake inhibitors. This is a category of drugs that many people your age are on. They're antidepressant drugs. And what they do is they allow the serotonin to reside in the synaptic cleft longer than it would normally. It prevents this recycling and then allows these downstream cells to have more serotonin. Well, so say this cell is involved in some pathway involved in the process. And it's sending a signal that is meant to get through, right? It's meant to get through. But the serotonin, it's released, but before enough of it can attach to the downstream neuron and cause an action potential, so whatever emotional processing signal is supposed to continue down the chain, and leads to feelings good, or like this thing happened, I respond to it, and oh, I feel great that like my friend just told me that they like my shirt, and so I feel good about that. Thanks, great, I love it too. Love the shirt, and so I get a little boost of serotonin from that, right? But if your neurons are recycling that serotonin too quickly, that emotional processing that's there isn't happening. It's not happening. And so the signal's coming in, but it gets kind of stopped there because the serotonin that's being released is either being recycled too quickly and is not allowing the signal to get a strong enough signal to get into the next neuron down the chain, or maybe you're not making enough serotonin for some reason, and it's allowing the small amount of serotonin that's there to reside longer so that a stronger signal can get through. Yeah, serotonin is a neurotransmitter that's associated with neurons that lead to like the reward system in the brain, or sense of well-being or happiness. People say you actually like is serotonin. So that's why they have re-up. Serotonin, it's complicated. It's used in a wide variety of, for example, there are more serotonin receptor neurons in your gut than anywhere else in your body. That kind of makes sense. Like you eat delicious food and like that kind of thing. But yeah, serotonin is used in a lot of ways, but one of the things. So I mean, when you are depression, it's a lot of other that often go along with it, not just the memory very well, right? People are like depressed. What you did that day, or it can be associated with paranoia, or it can be like shakiness, sleep. Serotonin is so the way you make melatonin, which is yeah, it's the emitter that basically get melatonin is tryptophan, acid, serotonin, sleep. Anyways, that none of that is on. One other way that calcium and all of this whole exocytosis process is triggered again. The key is calcium and how calcium acts as a second messenger. Calcium is what's called a second messenger in the cell. You can have a signal from the outside of a cell come to the surface, and then it's going to cause a spike in calcium, and calcium can trigger all kinds of things like exocytosis, whatever. There's a lot of things. We haven't talked about how muscles work, but muscles work because there are these motor proteins that when calcium binds to the motor protein, it unlocks the active site, and calcium is the trigger, is the key that unlocks muscle contraction. Calcium is very important. Calcium, yeah. Calcium is super interesting. We could talk the whole rest of the year every day. If I lectured every day just talking about the ways that calcium is used in the body in detail, we would fill the calcium. You're getting like the tiniest little snow off the top of it. So exocytosis, muscle contraction, neurotransmission, and clotting are some important examples of how are there any other? I don't want to get too lost in it because I could talk about it for a lot longer. But and now on to Mars's question. So Mars wanted to know about osteoclasts. And I'm going to talk about all four of them. So there are two general categories. There's osteoclasts and everything else. First, the osteoclasts. These are it's like, osteoclasts are like the cell that ate Philadelphia. Easy horror movie in the 50s. It's that ate Cleveland. This is enormous cell. They're enormous cells that have many, many nuclei in them.
Osteoclast.
Yep. Yep. You don't need to know those facts. What you should know about them is that they are the bone-dissolving cells. They release acids and enzymes that dissolve the mineral content away from bone, which is important if you are, so you're gonna, right now I live in Waterville and they're renovating downtown. They, hotel down to tavern. And to build that, they had to tear down the old Levine's building. So if you're gonna build something new, in response to the need, you have to just break things that were there apart that are not useful as well. So osteoclasts are osteocorroding or bone corroding dissolved cells. And then the other category of cells, it's really a lineage of cells. The first of them is these osteoprogenitors. Osteoprogenitor cells are cells that are able, they are activated by stress on the bone. So like weight bearing or impact, yeah, that kind of thing. Yeah, sure, sitting cross-legged. Or if you hurt a bone, it's going to stimulate the osteoprogenitor cells to divide. And they live in this cellular layer that's coating both the inside and the outside of periosteum on the outside. So there are these cells. Important thing about them is that they can divide. It's the only stage in this where you can get more of them. You need more bone cells. It's up to the osteoprogenitor cells to divide. They go through mitosis, cell division, and give us more bone cells. Then they can differentiate. So they go through some changes. Certain genes turn on, other genes turn off, and they evolve and differentiate and turn into osteoblasts with a B. Osteoblasts are bone-building cells. And they make this stuff called osteoid. Osteocalcin is part of what's called osteoid. Osteoid is essentially the organic compound of meat. It's got the collagen, the proteoglycan, the glycosaminoglycan, the different organic compounds. We talked about bone matrix. I talked about collagen and proteoglycan and glycoprotein and glycans. All those like weird sounding words that were made of various proteins and carbohydrates. That is in the osteoid that's made by the osteoblast. Yeah. You have, I think, like 21 different genes in your body, and those different genes are expressed in different tissue types. So the collagen that's in is 7. Well, there's a couple different collagen, but that collagen, these different isotypes, is you can imagine it like different. They're all iPhones. They're all the one in your bone is a little bit different, but not dramatically so from the collagen. That's right. Yeah. They make the osteoid, and if the organic part has been made, then that's going to eventually turn into bone. All right. Yeah.
So it has like a
osteoblasts.
Yeah.
No, osteoblasts have one. They are a stage. There's 3 stages. There's osteoprogenitor cells, osteoblasts, and then osteocytes. They're all derived from the same cell, but they're just maturing. So you can imagine it like this. Say you go to college, and you are a freshman in college. You don't know what major you're going to. You're not sure, but you become a sophomore, and you start taking classes that lay down the groundwork for your degree. Right? You take your, English Lit class, basic math class, or whatever, and then you decide that you want to be a film studies major. All right, and that would be like you in the beginning you could have been any major, but you're like specializing now and you're honing down this thing. So osteoblasts are making osteoid. They're laying down this foundation. They've taken their general lit class or whatever, and this osteoid begins to surround an osteoblast. And once an osteoblast has surrounded itself completely with osteoid, then it goes through another transition and becomes an osteocyte. It's an osteocyte. And again, when it becomes an osteocyte, certain switches turn on, other switches turn off, and it begins to mature or develop.
Like one.
It's so this cell at no point dies.
Right.
It just changes and becomes this. This is what differentiation is. So, for example, you can have at conception, you have the egg from the mother and the father come together and begin to divide. Those first cells, they are not. There's no cell in your body that. Those are embryonic. Every cell in your body, they could go in any direction. They go in any direction. You turn the right switches on and the right switches off as they go along. And that's what's happening here. This osteoprogenitor cell, there are still some switches that can turn on and others that can turn off. And so there's these 2 other stages that it can go to. Osteoprogenitor cells, their primary goal is to divide in response to stress. Then the osteoblasts, it's to lay down osteoid. If you lay down osteoid, it's eventually going to turn into bone. So the osteoblasts build the cell up by adding osteoid, and then once they're surrounded by osteoid, that is a cue for them to have other genetic switches come on and off, and they transition into being fully mature bone cells. So neither osteoblasts nor osteocytes are mitotic. They're not able to divide. That is back here. These 2 are just concerned with building and maintaining bones. Osteocytes, they're going to calcify the osteoid that had been laid down. That organic matrix that the osteoblasts built, the osteocytes calcify that with hydroxyapatite. Any other questions? All right. So here is just that same concept. A small part of the bone is made of cells, the osteoclast, blastocytes, and progenitor cells. About a third of the weight is made from the organic stuff, like the collagen and the various carbohydrate protein things. And then about 2/3 of it is made out of inorganic hydroxyapatite, which is primarily calcium, phosphate, and some calcium carbonate in this sort of ceramic structure. No? Mm-hmm. Yep. Yes.
Yeah, I think it's like a.
So imagine Maynard's. Yeah. And if you were to take all of this school, the whole school, and put it on a scale and weigh it, what would it weigh?
Like
At
at least.
weigh a lot, right?
Yeah.
The people are the cells. The people are the cells. Only a little bit. Only a little bit of the weight. The organic matter would be maybe like the paper, this stuff, right? The plays that we're reading, the tests in the textbooks, and then the inorganic matter is going to be this is what gives it flexibility, right? This is where the flexibility. This is you're not learning from this. It doesn't have any information. It's just a solid thing. A chair, it's a solid thing. Floor, a building, all of that, solid stuff. So the matrix of the school is the combination of this stuff, the piano and the chairs and the buildings, and all of that. The cells are us. It's easy to think, oh, school is us, right? It's made of people, but there's this matrix that we live in that gives it a totality and holds it together, right? Like if we didn't have tables and chairs and this and that and the other thing, we wouldn't. Yes.
So.
Yeah.
Do it. It supports a or no, it can change.
Well, all of it changes. I mean, all the stuff in here changes. People,
Right.
shred stuff, and we get new stuff, and it regenerates.
So
right.
It's like it is
Mm-hmm. Yep. Yeah. Yeah. It's a good point. So I don't want to get too deep into piezoelectric materials with you, but it's actually a combination of both. It's a combination of. So it's easier to understand with the the collagen, and it happens in the collagen, but it also happens, in fact, even more significantly in the inorganic matter. Real piezoelectric material, when it's typically inorganic, when you compress a piezoelectric material, you are, I'll just say it in the actual, you are deforming the electron clouds shell atom in that material. By doing that, you're enabling electrons to flow through that material more readily. You're lowering to it, so you it can create an by, or at least a field by piezoelectric material. Not all piezoelectric, or not all materials piezoelectric. Turns out that hydroxyapatite is. And so, yeah, it's both the collagen and the stuff, and the hydroxy. Good looking. Yeah, anyone else? All right. So then I went on to talk about the homeostasis of calcium. And it's regulated by 3 hormones that are broken into 2 categories: calcitonin and then calcitriol and parathyroid. Calcitonin is the hormone that is in charge when your calcium in the blood is too high. All right. So the regulated variable here is what? What is the regulated variable?
The blood.
Yeah, calcium in the blood. That's right. And calcitonin is around when it's too high because it wants to tone it down. And the other 2, parathyroid hormone and calcitriol, also known as vitamin D. When I say vitamin D, I mean calcitriol. When I say calcitriol, I mean vitamin D. These are when the blood doesn't have enough. It's too low, all right? And you need to raise the calcium. This is regulated in 3 tissues, all right? And I should say that calcitonin comes from the thyroid. Parathyroid hormone comes from the parathyroid. And calcitriol, it involves 3. It involves the skin, the liver, and the kidney. Get off of her. Get off of her. Please. All right. So it's regulated in 3 tissues: the bones, the digestive tract, and the kidney. And the bones are where we store the calcium, the GI tract is where we absorb it, and the kidneys are where we remove it from our body. So this is the doorway in, this is the doorway out, and this is where we keep it. Yeah. I'll pull that slide up and hold on to that question. So on the one side, when the calcium is high, when calcium is high and calcitonin is being secreted, it causes the body to store calcium in the bones. It causes the calcium we eat to pass through your GI tract without being absorbed, and it causes us to pee more calcium out and excrete it in our urine. On the other hand, parathyroid hormone and vitamin D together, they're going to dissolve calcium and encourage osteoclasts to work more and osteoblasts to work less. So we'll dissolve more calcium, we will absorb more calcium in the gut, specifically because of the vitamin D, and we'll retain more of the calcium at the kidneys. The kidneys will not filter out the calcium in our blood already. Does that make sense? Any questions about that? Good?
Calcium is go.
If you are eating a ton of calcium, let's say you're taking... Yeah, usually it'll just pass through the colon.
So like better to.
Sure. Well, if you absorb it, it'll pass out through your urine. And if you don't absorb it, it's just stool. But if you have too much calcium in, like say you're taking calcium supplement, it can lead to, if you have like a whole lot of. So this is that. These are the two loops of the calcium homeostasis. This is the loop when it's too high, and this is the loop when it's too low. So this is going to be the one that's dictated by calcitonin, and then this is the one that's going to be dictated by parathyroid hormone and vitamin D. We'll do this one first. Very high calcium in the blood stimulates the thyroid, releases calcitonin, and causes its effect on the bone and kidneys, and the blood calcium declines to the set point. All right, now say the calcium in the blood goes too low, it's going to stimulate the parathyroid hormones, releases the parathyroid hormone, makes the calcium come out of the bones, and then at the kidney, it makes the kidney resorb more of the calcium from the urine than it would normally. And at the kidney, it also activates vitamin D. Vitamin D is not active until it's active by the kidney under the direction of parathyroid. We had a slide about that. So then it's vitamin D that has its effect at the gut and allows you to absorb more of the calcium. One of the points here is that we have all these organs involved, right? The kidney, the gut, bones, skin, etc. But the most important organs for this whole process are the thyroid and parathyroid. Those are the ones that are sending out the signals, controlling what's going on. So here is the vitamin D story. This is the answer to your question. So, yep, you have cholesterol that's in the cells of your skin, all right? And we have sunshine on the skin, and when that happens, those epithelial cells in your skin convert cholesterol into vitamin D3, into a type of vitamin D. That circulates around the blood, and when it gets to the liver, it gets converted to another intermediate called calcidiol. Calcidiol is circulating around the body in the blood, and when it gets to the kidney, if there is PTH around, then it gets converted to calcitriol. Calcitriol or calcitriol is the form of vitamin D that is active on the gut. Calcidiol, vitamin D3, and the 7D dehydrocholesterol, none of them have an effect on the gut in terms of the absorption of calcium. It's only after this intermediate is converted to calcitriol at the kidney, because if there's PTH, does this stuff, is it able to have its effect in the gut? So what's happening is in terms of the organs that are necessary for the absorption in the gut, you need the skin, the liver, and the kidney. All right? Skin, the liver. So they're different molecules. They are related molecules. They're being modified with time. And vitamin D3 has effects in the body. It does other things. Like vitamin D is related to mood. And when it goes to the liver, it's going to be converted to calcidiol. Calcidiol has its own effects in the body. It's doing other things. But calcidiol is also it's like a trigger, right? Or it's not a trigger. It's you have all this stuff on hand ready to make calcitriol, but it's like the inactive form of calcitriol. And for it to get activated, we need to have PTH in the bloodstream. If PTH is in the bloodstream, then PTH affects the kidney to convert calcidiol to calcitriol. I know this seems like a complicated set of dominoes, but take my word for it. This is actually very streamlined, very simple set of dominoes. I think I said when we were clotting, 35 intermediate steps in the clotting. This is just basic biochemistry. Super fascinating. It's amazing to me. I don't know. I think it's amazing that our bodies develop. Yeah. This is why I talk about that in class, where people in northerly climes are more prone to rickets, which is why they started adding vitamin D to milk. Adaptation. All right. So you can have osteoporosis. People can get osteoporosis with age. And we've talked about parathyroid hormone and calcitriol and calcitonin. But estrogen is also really important in regulating bone density, the female androgenic hormone. And it's because it has effects on osteoclasts and osteoblasts and osteocytes. It can actually the lack of estrogen, its effect through T cells and directly and through osteoblasts can not having estrogen can allow osteoclasts to be unregulated, and so they just start going crazy. And then when that happens, in women, particularly after menopause, their estrogen drops out, and it leads to osteoporosis, the dissolving of bones, right? Weakening of bones. These are some ways that people deal with it. You can go through estrogen replacement therapy, right? So if you're having estrogen replacement therapy, you're getting pills or whatever, but are slowing that process. You're basically tricking the body into thinking that it didn't go through. So this is common, estrogen replacement therapy. Then there are also drugs like Actonil and Fosamax that actively destroy their what are called cytotoxic to osteocytes. And then you can also get daily injections of parathyroid hormone. But that's a little weird to do because your body's probably already making plenty of parathyroid. You're like overdriving the system. And then the best way to not get osteoporosis, and it's no guarantee, but the best way is to just not get it in the first place. The best way to deal with it is to not get it. And that is to have like a good daily exercise regimen and eating healthy food that's appropriate for your body. I should pull it out of this slide because I don't really, that slide up there, it's pretty much, yeah. I just wanted something that showed that estrogen has an effect on osteoclasts, through, I don't want to go through it. It's the system that I've described for you is an oversimplification of the relationship between a lot of these players. A full description. All right. So, why did I have this to ask? But, yeah. So, this mineral recycling, a couple of these points are important. The mineral recycling allows bones to adapt to stress, right? So because we're taking stuff apart and we're building stuff up, the bone is able to respond to needs. So for example, this is like urban renewal in a town. Water bill, half away, goes down, away, stressed, and you can either, like the town can wither and die, or you can try to respond to that stress. Take structures that are no longer useful and apart and build new things there, or renovate things that are there, like use some of the structure that was before but change it. Heavily stressed bone becomes thicker and stronger. The more you use it, the more it's going to respond. So, for example, you can, like Manhattan, right? It's under this huge amount of stress. There's so many people that want to be in Manhattan. The more people that are there, the bigger you're going to build the buildings. The thicker and stronger the buildings become. I think that's points in this slide. And then there was the piezoelectric bone. So I talked about this a bit ago when Beatrix was sharp eye caught my not deception but oversimplification. But in collagen adds to the piezoelectricity by when collagen gets compressed. Say this is running along the length of a bone. So you can imagine, I guess, collagen is like this, right? In the osteoid, collagen fibers are like this. But if I'm if I'm pressing on the bone like this, the collagen that's laying along that axis, the collagen that's laying along the axis of compression, that is going to be opened up like this, whereas these fibers that are perpendicular to it will not. Okay, those will not. And when they open up, it's going to attract the osteoblasts to the collagen, and they're going to lay down more osteoid. And as they get there, they're going to convert to osteocytes and lay down hydroxyapatite as osteocytes. And so you can imagine that's how, say this is the line of stress. Well, this is going to become calcified here, and this collagen going in the other direction will not, right? And this is how we get to that. Wait a second. I want a picture of bones like that. This is how we get these stress patterns in bone, right? So you see these like lines along here. This was because stress, like this muscle was pulling on this bone this way, and this line of stress, the collagen that was along here, got calcified, and the collagen along here got calcified. The collagen that was running this way wasn't being stressed, and so it didn't get calcified. All right? Yeah. The collagen is part of the osteoid, right? The collagen fibers are there, and glycosaminoglycans get excreted around that, makes the osteoid, and then that gets turned into the hydroxyapatite. Yeah. Well, the matrix, the osteoid is getting calcified, and when it gets calcified, it is now bone. It's bone. Yeah, that's already like a deeper level of nuance than I'm going to ask on the test, but or assessment or super mega fun time or what do we call it? Supper mega fun time. Yeah. So okay, we're getting close to the end here, and it's snowing. It's really pretty. Exercise, hormones, and nutritions. Yeah. Vitamin C, vitamin A, K, and B12 beyond vitamin D. Those are also important. A quick take home. And last but not least, osteoporosis prevention. Exercise and diet between the ages of 25 and 40, 45 are see.
Last class is
Not specific. Healthy bone development. Okay. So all of those slides are up on the thing. I will see you on Thursday. There is going to be there are 11 multiple choice and 2 short answers.
Whoa.
You're done. Class is out. Get out of here. Yeah.